Hello friends, welcome to another video tutorial. Today we will discuss about the prokaryotic cell. What are the characteristic features of a prokaryotic cell or organism? First of all, we will see the structure of a prokaryotic cell, which is in front of you, and it shows that the prokaryotic cell contain a central genetic material in the form of DNA, which is known as nucleoid. Later, we will discuss about why it is known as nucleoid. Another structure nearby this nucleoid, a 70S type of ribosome. So this is the ribosome. And again, we see a small circular DNA nearby this DNA, which is known as plasmid or extra chromosomal DNA. Extra chromosomal DNA. We will discuss one by one in last. And then if we saw a small dot-like structures which contains food particles, which are known as the inculsions or food granules. Apart from this also contain a small vacuoles which filled with the gas, which are known as the gas vacuole, and a liquid filled inside the this prokaryotic cell is known as cytoplasm. And if we go outside, a structure which surround this cytoplasm is known as plasma membrane, and which is surrounded by a cell wall, and in some bacteria a small gelatinous layer also surrounded the cell wall, which is known as capsule. Apart from this, a small hair-like projections just like Rome is known as pilus, which also help in the sexual reproduction. We will discuss one by one. And a long thread-like structures which are known as flagellum or flagella, which help in buoyancy or in migration or movement of the bacteria. Let us discuss one by one. Before this, we must draw a small diagram. This is the bacteria, some because there's some structure are not present in this image. This is the bacterial. We will discuss one by one and we will draw the diagram one by one all the component. So first of all, we saw that this is the bacterial cell membrane, and this bacterial cell membrane shows some inward foldings uh, like this, and this structure which fold inside is known as mesosome. This structure is known as mesosome, and it contains small enzymes on its body, and these enzymes which are attached to its this mesosome surface are known as respiratory enzyme. So, very important question come from what is the function of mesosome and what is mesosome? If we saw that this mesosome function as an analogous to mitochondria, analogous to mitochondria, mitochondria, because when a bacterial cell divide, it enlarge itself and help in cell division. Second point, help in cell division. And in a eukaryotic cell, most of enzymes are present in mitochondria who perform the activity of respiration or formation of ATP from the complex. So ATP are not synthesized inside it, but it contains respiratory enzyme on its surface that was it is analogous to or somewhat it function like a mitochondria in this prokaryotic cell. So it will also known as analogous to mitochondria. Similarly, some outgrowths are also present on on this membrane outside. Some outgrowths from this membrane goes somewhat outside like this, and these structures are known as pilus or pili, यानी कि hair like structures, roams. Uh, so it is known as hair-like structures, which are known as romes, and they help in. It is present only in some bacteria, and the bacteria who contain these romes and the plasmid, which they are known as the male. They are known as the male, and who does not contain this pilus and this plasmid are behave as a female. So during a conjugation or as reproduction in bacteria, this pilus or pile help in attachment of this another bacteria who behave as a female to the male, and a copulation bridge or a conjugation bridge is produced in between them, and the plasmids one copy migrate to in this bacteria, and this bacteria now again both were separated, so this bacteria also produce pilus and have plasmid. So in this way, the one bacteria who does not contain contain pilus after conjugation. So this process is occur with the help of only this pilus. So it help in sexual reproduction. And this male, because it donate its plasmid to this who does not contain plasmid, so this is known as male because it donates its DNA to the this another bacteria who does not contain this plasmid. And what is the plasmid? Plasmid is the extra chromosomal DNA. What is plasmid? Extra chromosomal DNA means the another copy of a DNA which is present in bacteria. It is circular and it contains genes for sexual reproduction. It contains gene for sexual reproduction, and it contains genes for antibiotic resistance. Antibiotic resistance. So both genes are present on this plasmid, and it is present in bacteria like a circle. Similarly, if we saw the bacterial DNA is present like this, so it is known as nucleoid. Why it is known as nucleoid? It is a very important point because in a eukaryotic cell, if we saw that their DNA is covered by a membrane or in a nuclear membrane. But in this case, the DNA is directly present inside the cytoplasm, the liquid filled inside the this bacteria cell. It is known as cytoplasm, known as cytoplasm. And this DNA is present only in cytoplasm, and no envelope is present around it. So, in this condition, it is known as nuclear. Nuclear means anything which behave like nucleus. If a oid word is attached to anything else, so it's lose its meaning of front word. For example, nucleoid mean looks. It is not a nucleus, but it behave as nucleus. It's not looking like a nucleus, but it behave as a nucleus. So this condition is known as nucleoid. So the bacterial DNA is naked. Very important point. The bacterial DNA is naked, or we can say prokaryotic cell have naked DNA. The word prokaryote, pro mean primitive and karyote mean nucleus. Karyote mean nucleus. From this, the prokaryotic word is come, which means primitive type of nucleus. So prokaryotes mean primitive type of nucleus. Okay. So what is primitive time? The primitive type is that the DNA is not surrounded by any nuclear membrane and the histone proteins are absent. Histone proteins are absent from DNA. In higher classes we study, or in another class I will previously discuss about the histone and chromosome. So this DNA you can saw the video in my previous videos where we will discuss that there is a histone protein octamer on which DNA is coiled like this, take two round and migrate to another, then another histone. So DNA is coiled around the histone proteins. So in prokaryotic cell, this histone octamer is not present and a nuclear membrane is not present. That's why it is known as nucleoid. Okay. Another structure that is known as 70S type of ribosome. It is important for the prokaryotic cell contains a 70 type of S ribosome. Ribosomes are known as the protein factory of a cell, and they help in protein synthesis, or they perform the protein synthesis. That's why they known as the uh, protein factory of cell. So sometime in some bacteria you also saw that there is a like structure, a structure like this. This is known as polyribosome. Means this is the mRNA on which many ribosomes are attached and perform the function of protein synthesis. In such condition it is known as polyribosome. Okay, it is known as polyribosome. uh, Another structure that is known as granules, like some food granules are present inside the bacteria. When the bacteria have a sufficient amount of food, it stores some food in the form of glycogen glycogen granules. So it store foods in the form of glycogen and utilize it when there is a scarcity of food, or sometime when there is a no food for bacteria, then it utilizes this glycogen. Okay. Some bacteria also produce some granules in the form of oil. So in some bacteria, oil is also present. It is depend upon which type of condition bacteria is living. Then a gas vacuole, I draw like this, a structure is present on which a gas or air is filled. This air filled vacuole help in bacteria to buoyancy. Buoyancy mean help in moving on, help in moving over water or in air. It help in swimming, swimming of bacteria in air and water. Okay, so it has some bacteria contains gas vacuole. That's why bacteria present omnivoreously. So these are the some internal structures. Let us discuss the surrounding structure that is known as the cell membrane. Like other cells, bacterial also contain a cell membrane, and the cell membrane is mostly made up of three things are present in a cell membrane. And what is these cells? That is the fat, which is present in the form of phospholipids. Second is the protein, and some proteins are also present in the form of glycoproteins. So some proteins are in the form of glycoproteins, some proteins are freely present, just like another cell membranes. So we will discuss cell membrane separately in another video when we will discuss about the eukaryotes and their cell organelles one by one. So we will discuss this proteins and cell membrane and phospholipids in a next video. Hope so. Later on. So for that we must know about the proteins, fat, and some glycomins, carbohydrates. Carbohydrates are not present in freely, they are either attached to the fat, then they are known as the glycolipids, or they may be attached with protein, then they are known as the glycoproteins. So these are the proteins which are present in cell membrane. Now discuss about the now a cell wall is present which surround this structure, and some structure like this are also present in some bacteria. These are known as the flagella. So first of all, we will discuss about the cell wall. In a prokaryotic cell or bacterial cell contain cell wall which is made up of mucopolysaccharidyl glycan. As the name describes, mucus, polypeptide protein, glycan means lipid, or simply it is known as peptide, peptidoglycan, peptido, peptidoglycan or peptidoglycan. Okay, or commonly it is known as murine. Commonly known as murine, so we can say it mucopolypeptide glycan, peptidoglycan, or murine. So cell wall of bacteria is made up of murine or peptidoglycan. And these some structure which are looks like this, present in cell wall, and a hook-like structure is also I draw on our is present in this. If we look at the diagram, in this diagram also I made it like this, and if we go to this, this is the this it shows the here a hook-like structure is present. So what is this? Let us discuss it. This is the base. This is the base, and this structure is known as hook, which embedded inside the cytoplasm of the bacteria. And in this a filament, which is made up of prokaryotic fibers, or type known as the prokaryotic flagella, made up of fiber. These fibers are known as the proteins fiber known as the flagellin protein. Very important, the bacterial flagella is made up of flagellin protein, which is a primitive type of structure in which 9 plus 2 arrangement is not found. Arrangement is not found. Not found. Very important word. Not found. So this flagella, this structure is fixed, and this structure, this rotate in upward, backward, forward, in any direction, and also rotate centrifugally. So it produces a gyratory motion. Gyratory motion. This gyratory motion help in bacteria to move in upward, backward, forward in every direction. just like a boat. If you travel inside a boat, you have seen the chappu, a instrument used to move the boat in water canals. So this flagella help or behave like a chappu to move a like this key like move forward as we push the pressure by the chappu inside the water, the boat move forward. If we use this in like in this situation, then boat move in this direction. So similarly, this bacteria contain flagella. If these flagella move in this direction, all flagella move in synchronization, then bacteria move in this direction. If these flagella move in this direction, so bacteria move in this direction. So they help in backward and forward, upward, downward movement of the bacteria in any direction. Bacteria move with the help of these flagella. These flagella are made up of flagellin protein and help in movement of bacteria. Simply one thing you must remember, help in movement of bacteria and they shows a gyratory motion. Okay. Now, last point which is very important, that is the presence of a thick xylem layer or sometimes thin xylem layer around the bacteria. Uh, like this. This is the capsule, which is known as capsule. This capsule is made up of a gel-like structures, gel-like structure or jelly, mucous membrane, like a mucous membrane, and it protect the bacteria. It protect the bacteria. The capsule formation is not the character of all bacteria. Only some bacteria produce the capsule around themselves to protect themselves from unfavorable or desiccation. Unfavorable conditions to protect themselves from unfavorable conditions. Some bacteria produce a slim layer around themselves. For example, a bacteria which is very popular in India, Clostridium botulinum. Clostridium botulinum. It produces a capsule around itself and can tolerate the temperature of 300 degrees Celsius. And what it do in India, in somewhere in midday meal, and we saw a news there is 220, 200, 100, and many a group of peoples die due to food poisoning. So maybe this bacteria maybe grow there. And when the food is prepared and heat up in a temperature, it will not die because it survive at 300 degree Celsius temperature and it produce a toxin known as botulin. This botulin cause death of individual. So some bacteria produce this capsule. It is not the character of all bacteria, and it help in desiccation or tolerate the unfavorable condition by the bacteria. One thing you must remember here, if the capsule is present on around the bacteria, so then this bacteria may lose its flagella and pili. Similarly, I already told that the pilus is not the characteristic feature of all bacteria. Only some bacteria are contain pilus and this plasmid. If pilus and plasmid is present on a bacteria, then this bacteria is an act or behave as a male. It act as a male if these two structures are present to it. Uh, okay. Now, what is this? Fimbri or pilus are the same thing, so don't be confused. Flagella made up of flagellin protein, which help in movement of bacteria. Plasmid is the extra chromosomal DNA, which contains genes for antibiotic resistance and sexual reproduction. Inclusions, food particles or food storage granules, which are known as cells. Nuclear DNA without histone proteins, and a nuclear membrane. Ribosome 70S type, which act or behave as a protein factory for this bacteria and produce protein for this. Polyribosome, the ribosomes are in the active form to producing protein. Cytoplasm, the liquid filled inside the cell known as the cytoplasm. Gas vacuole help in buoyancy in water and air. Cell membrane made up of proteins and lipids and some carbohydrate which are known as the glycoproteins and glycolipids. Cell wall made up of murine or peptidoglycan. Uh, okay, so I think so you will understand everything. If there is any doubt, kindly let me comment. I will discuss in next video, and kindly subscribe our channel, like video. Thank you so much. Radhe Radhe, Subh Ratri.